So hello. So first, so that I don't forget, here are some reviews on the subjects of the lectures we did so far and today. So first, on evolutionary rescue in general, there was three quite recent reviews. There was a special issue, a whole special issue on the subject, then two recent reviews, or recent. Yeah. There may be more recent reviews. These are the ones I know of. Also, some reviews on the Luria-Delbruck experiment and distributions, mostly on the theory that goes with it, because it's a rescue, and sometimes it's fairly disconnected, those two literatures. So Hélène Alexander connects them in this review. But yeah, it's interesting if you're doing experiments with bacteria. It's interesting to read that, I think. And today I'm going to talk about a phenotype fitness landscape, which is the most classic one, which is called Fisher's geometrical model. And there's a review on it too in 2014. So let's get back to ER. So ER will be evolutionary rescue. So just to remember, we stated the first possibility for rescue, which is that you have rescue from a single step, meaning that from the wild type that decays in gray here. So this is size versus time. The wild type decays, and from it, some mutant arises from this wild type and has some stochastic growth initially and then makes it up. So it's a resistant mutant that establishes. So during this phase is not extinct and goes up. If it's born during decay here, it's a de novo one-step rescue. And if it's born before decay, so it was there in the population initially, it's a standing variance rescue. So the basic models I showed you where the standing variance is at mutation selection balance. You can find it in a nice paper by Orr and Uncles. In general, I like the way Orr writes. So he served as my reference in PhD on Fisher's model too. So it's quite nice to read, and it explains the basic models that are here. And they don't consider multiple types possible or other demographies than this. It's a discrete time model, but it covers this first problem. So here is what happens. Yeah, when you have this is what you see as the population size decay and then growth back. Now, another thing can happen, that rescue happens in two steps. So what does it mean to be in two steps? It means that you have a first mutant that will not make it to establish and rise to fixation. But this mutant produces secondarily one mutation, which is therefore two steps away from the wild type genetically. So there is a de novo two-step rescue, which is with so we call it springboard mutations in another paper, not this one. In this one, we do the population genetics for it. They can be called springboard mutations because they serve as a springboard to become the rescue. So either this springboard mutation arises de novo, or it's already there and it serves as a rescue later on. So to be a two-step rescue, you need this springboard not to be a rescue. So it may be a resistant mutant that did not make it to establish, or it may be a non-resistant mutant that will never make it. But what's important is that it's a second step. Ideally, experimentally, we should be able to disentangle the two. I'm not fully convinced from my experiments. I think there are other ways to know, because we should see something first decay and then something a bit flat and then the rise. The problem is that sometimes the decay is so large that you go into your blank signal and you don't see what's happening. So you don't see if it's a triangle, if you want, or a basin like this. So there are other ways to look at it. I can discuss that if you're interested. And so this is two-step. But now, all this is considering just one mutant type. There's always, in every replicate, there's always the same mutant that may or may not arise, but it's always the same mutant, has a given mutation rate to it and a given growth rate and a given set of demographic parameters. But of course, there are several possible mutants. And the other thing that we would like to put in the soup is a connection between the type of stress you impose and the likelihood of your mutants being resistant, and also their growth rates. So the mutation rate to resistance versus the stress you put, the growth rate of those resistance, and also the cost they have in the past when it's relevant. So we're going to talk today about the extension of this to have various possible types and a simple model to derive those mutant types versus stress level, which is a phenotype fitness landscape. So what is this principle of this landscape? Here I'm plotting a phenotype, some phenotype, which we don't need to detail, in fact. What is important about this phenotype is that it's under stabilizing selection. So there is an optimum for this phenotype. And the curve here, blue, shows, well, sorry, the first curve to see is the dashed curve. It shows the growth rate of this mutant, of the phenotype versus the phenotype. There is an optimum here at the corresponding to the maximum growth rate. This maximum growth rate we may call R max. And mutations happen from a wild type in black to become a mutant in red. They can go one way or the other. They are symmetric about the wild type. And a change in the environment there is conceptualized as a shift in the optimum in typical literature. So this is really a central model of evolutionary ecology when you want to deal with changes in the environment that you want to characterize. For example, the environment changes over space. So you have the evolution. This is really the niche kind of concept. If your environment changes over space, you have models of, for example, the area, the range of the species based on that. Like when you go away from the native place of the species, the environment is less and less good. So the optimum is in fact farther and farther. And so there are no genotypes that are able to survive in these far away environments. You can do also a variation in the environment in time, where the same thing happens, except that now it's the phenotype that the function here shifts over time, so that your wild type who was happy initially becomes maladapted as time goes on. So there are 2 or 3 important original ER models that they are not the first ones, because we saw that Gloria Delbruck did rescue models, but they are the first ones dealing with this characterizing environmental change in terms of something that has a meaning for the genotype effects, for the resistance probability and all that. These models are all deterministic, and they are in sexuals, where when in some conditions, at least with weak selection, we can assume that the variance in the population of the phenotype here remains constant. So the response is really a traveling wave in phenotype, which responds to, well, it's traveling wave, not traveling at constant speed because it stops at the end, but it's a shift in the distribution of phenotypes that can follow the change in the environment. So there are models that consider a single abrupt change. So you go from here in one infinitesimal time, you shift to this new environment, and your distribution of phenotypes has to move for adaptation to proceed. And initially, so you have decay because your distribution is there. And as you adapt, you go down and back up in population size. The other 2 models that have been derived early on were models where instead of an abrupt change, you consider a linear change. So meaning that the optimum moves at a constant speed over time, but it never stops. It goes on and on and on. And there also, there are some nice simple outcomes regarding whether rescue can happen or not, depending on the speed at which the environment moves. Basically, if it moves too fast, the population cannot follow and it dies out. And if it doesn't, the population will die out certainly. So these are deterministic and sexual models. And of course, for a problem with bacteria, and in general, it would be nice to have a stochastic treatment of this problem and also derive it in asexual. So what is special in asexuals is that we cannot assume that this smooth polymorphic distribution of phenotypes will be there in the first place, and more importantly, that it will be responding at a constant speed given by the variance, well, depending on the variance of the standing variance. So we have to work out our individual mutant stochastic models that we saw in the 2 last lectures, but we have to work them out in this particular context. So here is just an illustration of the parameters of the model. Here we have to be more precise, like it was the case also in these former models. It's not just symmetric mutation and some optimum. We have to be characterizing the shape of this and the distribution here. So the assumptions are that the growth rate, which is not well drawn here, it would be exponential of the growth rate here. The growth rate is a quadratic function of phenotype in multiple dimensions. There's no problem with dealing with any number of dimensions. And the mutant phenotypes, they are normally distributed around the mean, around the parents, asexual parents. This normal is isotropic over space. There are ways to do anisotropic, but it's more complicated. And it has a given variance. So lambda here is really the variance of the mutation effects on phenotype. There is a mutation rate, which is considered constant at the genomic scale. And we have Rmax, which is the maximum fitness in the new stress environment. Lambda is this step size, n is the number of dimensions, how many phenotypes are under stabilizing selection. So what happens is that you start with a wild type that had the former environment was optimal for the wild type. You can assume that the wild type is somewhere there, or that the population overall is at stationary mutation selection balance for this past environment. You shift the environment, and in this new environment, you're now looking not at relative fitness, but absolute fitness, because that's what counts for rescue. So there is kind of an iceberg out of the water, and the water level is the zero fitness, zero growth rate. And whoever is in phenotype corresponding to a positive growth rate is a resistant mutant, and whoever is not is a subcritical mutant, it's not resistant. And now the shift that is imposed by the change in the environment determines, so the shift in terms of phenotypic optimum will determine the decay rate that you obtain, because initially you were like here, if you want, in 3D, and as the environment shifts, boom, the wild type drops to a lower level. So here are the quadratic fitness function and Gaussian mutation effect. So what's the point of this is that by doing so, you make a link that emerges from the model between the distance to the optimum, which is characterizing the stress intensity in terms of phenotypic space, the decay rate, of course, because the farther you move, the higher the decay rate, which is an important parameter of our rescue models so far, and also the genetic basis of resistance, the proportion, the growth rate, and the cost of the rescuers, because of course, there are less and less random mutations that manage to be in the green area if you move this green area is further away from the wild type. So it produces, in one model, the different ingredients we need to plug them in our rescue models that are depending on a given distribution of cost, of growth rates, covariant distribution, etc. So
question. Yeah. I think I don't understand why is this asexual population? Where is that asexuality coming in this? Where is that assumption?
Oh, yeah. In what I'm describing right now, there's no assumption of asexuality. It's just the genetic basis of the relationship between phenotype and fitness.
So right now is this usual stabilizing selection model, right?
Yeah, it's a stabilizing selection model. And so it's not, it's been used in sexuals first. So all those models the papers I was citing here, they use this kind of model, but they are in a sexual context. And so they have the reason to do that was that it allows you to connect those different things, the stress level and the genetic basis and the decay rate. What will be critical in this asexual assumption is the way you will respond to the change, whether you're a sexual large population or you're an asexual population will be different in terms of the response you have. So
that hasn't come yet. Okay, so we'll wait for that.
If we were doing sexuals, this distribution of red dots here would not represent mutants. In most of the models I know of, like the past ones, this is really a stunning variance of it's a stunning variance that is there in the beginning, and there are infinitely many types, and all types exist at the onset, and they will all remain there. What happens is just how many the fitter type will go up, and they will again mix with each other. So this distribution of stunning variance will evolve according to typically the Landy kind of model. of selection of quantitative genetics. Here we cannot do that because we have no sex. We typically have no variable population. So we have to worry about single mutation events happening here. So that's the way we construct new genotypes. That is different. So that's the way we will respond to genetic to the stress that will depend on being asexual versus these sexual models. But if you're interested in the sexual models, really the original ones are there. They are analytic and they deal with the problem. They are not stochastic.
Yeah, no, I am aware of that literature. I actually have worked on those models. So that's why I was trying to differentiate how these differ from the asexual ones.
Yeah, they differ by the fact that they don't put any mutation process. They just respond from standing variance.
Yeah.
And this standing variance is like infinite, non-limiting. You have and it's also Gaussian. So there's a Gaussian distribution of phenotypes at all times. And this requires an infinitesimal model plus a weak selection.
Right. Yeah. Yeah. No. Those ones I know. So yeah. Okay. So let's see the asexual ones.
So here we will not make the assumption of first of infinitesimal model, of course, because there's no recombination, and we will not necessarily, depending on the case, we will not necessarily assume weak selection. Okay. So we have this connection, and now what do we do? Well, we use a set of results that we have on what happens to the mutant, the distribution of mutants under this model, the mutant growth rates. So first, there is an explicit distribution for the distribution of the growth rates of random mutations, so not just resistant, all the possible random mutations. We have an explicit distribution for this growth rate distribution, depending on the wild type, the parents' growth rate. So that's useful because we can connect how, depending on how maladapted the wild type is, what distribution of growth rate it produces. So it depends on the parameters of the model, n, max, lambda. Second, we have an explicit treatment of the mutation selection balance in this case. So it's not a mutation selection balance under sexuals, it's an asexual mutation selection balance. And it's not fully explicit, or it is explicit, but what we use is two extreme scenarios. So either strong selection, weak mutation, where you have a lot of mutations and they are not selected too much in the past environment. So then in this case, you can show that there is sorry, I'm saying strong selection, weak mutations. So I'm saying the opposite. So the red would be strong selection, weak mutations. So mutations are rare and selection on them is strong. So basically you have a cloud that is spread at every mutation and you have your fitness function that is narrow. In this case, there is a spike at the optimum and the weight of the spike is known depending on the parameter. It's proportional to E minus U.
Yeah, so Guillaume, I'm confused a bit. So N is what? This is the usual Fisher's geometric model? Is it this N dimensional?
Yeah, it's the number of dimensions at Fisher's geometric model. So N is going to be the number of dimensions. Lambda is step size, meaning it's the variance that is produced by mutation on each trait. R max will be the peak height, how high you go in absolute growth rate. U would be a genomic mutation rate. And I'm not going to be talking about per division rate versus not. So basically assuming that there's a fixed birth rate, if you want. Most of what we did were based on discrete time models. So I don't want to go in details of actual bacteria application, but basically it's going to be the same if the birth rate is given. So we can discuss that in the end. If the birth rate doesn't change, the wild type birth rate doesn't change. And RD is going to be like in the talks we had. So it's going to be the decay rate of the genotype. So we know the equilibrium distribution in strong selection, weak mutation balance. It's basically very spiked. There's a spike at the optimum, and then the distribution is spread around, not Gaussian at all. And overall, we have a distribution of all the types at the stationary state of mutation selection balance in the past. Under a weak selection, strong mutation case, so now it means that the fitness function is quite wide and the mutants have a narrow distribution. Then there is a kind of central limit theorem that comes in, which gives a result comparable to Landy's result on mutation selection balance in sexuals. It's basically, well, it was like, so I asked Ruth Landy, it was conjectured in his work on multivariate mutation selection balance. Here we just see it as emerge as a limit for a parameter. And the parameter being small is this, the square root of mutation rate times mutation effect. But that's the Landy's model at mutation selection balance for a single locus. And so in this case, the phenotypes are now Gaussian. So they are centered on the optimum again, but around that, there's no spike. There's a Gaussian distribution with a variance mu, where mu is this thing. Well, whereas standing variance, mu, sorry. Standard deviation. No, that's mu. That's mu, sorry. Anyway, there's a Gaussian with a given variance given by this. So we can come up with kind of a heuristic threshold for when one regime happens and the other. And it depends on the ratio of mutation rate to mutation effect, which as expected. And this ratio, whether it's below the dimension, the half the dimension to the square, or whether it's way above it, you have a spike and a non-Gaussian distribution of phenotypes. Otherwise, you have a Gaussian distribution. So we have kind of a quantitative threshold to know in what situation we are. So these are known, so we can use them. And in the weak selection, strong mutation regime, and only in this one, we also have the expected trajectory of mean growth rates over time as you adapt after a shift in the environment. So here, that's what would happen if you are in this regime of a strongly polymorphic regime. So well, weak mutation, weak selection, strong mutation. Mu is our parameter here, the square root of u times lambda. Rmax is the peak height. Rd is the decay rate. And that's what would happen in a deterministic model. So it means that it's the expected fitness dynamics when there is no effect of drift, of stochastic loss, etc. So it's in a large population that you shift. This gives you your trajectory of mean fitness and your rescue trajectory. But it's not very interesting for rescue because rescue is certain. You just will go down and back up. But it gives us an expected trajectory for mean growth rate. So that's the tools we have for this.
Yes, so sorry, but here you still have demography, is it?
Yeah, I'm just, for the moment, I'm saying what are the tools we have on this model.
Okay.
Because we're going to use them for the rescue model.
Okay.
Then the other tool we need, this is all distributional distributions of growth rates. What we need then is now we cannot have the comfort of saying we don't say anything about the stochastic demography, because if we want to give explicit results depending on the parameters of the growth rates, etc., we need to be explicit about how these parameters will affect the stochastic demography of the process. So what we used is an arbitrary, it's a diffusion limit to arbitrary density independent branching process. And this diffusion is known as Feller's diffusion. So instead of considering either continuous or discrete time, so these are figures from this text. Well, it's a paper, but it's really, to me, it's more like a textbook that has a lot of information. So really read it if you're doing stochastic demography or genetics. It's very full of useful tips. So this, you may have models that are discrete in time, so that generations don't overlap. You may have models that are overlapping, like the birth-death process here, and this is I guess a branching process, discrete in time. But in both cases, those two models, if the change is the expected change in log size of your lineages are small compared to the variance in the change in log size, you can take a diffusive limit to the size of the population. And this diffusive limit goes from either discrete or continuous time to continuous time directly, and from discrete states here, by definition, these are counts of guys, so it's discrete, to a continuous state. So the number of individuals is is treated as a continuous real number, positive real number. So that's a diffusion approximation. It's really, it's by Feller, it's a 51 paper, and it was intended initially for genetics. It's also the simple model for population genetics for frequencies. So it requires density independent growth or decay, and it requires weak effects and rates, just like the diffusion limit in population genetics. So we're going to use that. Why? Because it avoids saying too much details on the actual demography. So whenever we're in this general context, we can use the result, but we need these weak effects assumption. For example, if the population does huge jumps in an infinitesimal time, you cannot use this diffusion. You have other diffusion limits, but you cannot, because you need basically dn to be proportional to dt. So every dn. So if dn does a jump of 100 over dt that is infinitesimally small, it doesn't work. So for example, with viruses that have rare big burst events, it may or may not be a problem. It's much more demanding to have a virus demography being modeled correctly by a diffusion. Okay, so we're going to use this tool. And what does it boil down to? Well, you have a stochastic differential equation to describe your population size. I'm just checking the time. So let's say NIT is the density of genotype I at time t. Ri is the growth rate of the genotype. So this is an intrinsic property of the genotype. It may vary deterministically with time, and there are results. It doesn't change the level of results we have, basically. We won't use, in this lecture, we won't use the fact that it varies, but we will. So yeah, we will. And there is a sigma, which is the variance, the reproductive variance. So really, it's the variance of the change in log n. It's not the variance in n. It's a change in log n has a given variance over dt, and this variance is given by this. And R is our classic growth rate, which is the expected change in log n. So if you have these two things defined for a given genotype, sigma might depend on I, but in our models, it's going to be common to all genotypes. That's important. You can write the change of dn over some infinitesimal time as Ri ni dt, which is really exponential growth, deterministic, plus sigma ni square root dbt. So dbt is really, it's an infinitesimal change that is distributed roughly, although I think Amaury would hit me if I said that, but roughly you can consider that this thing is just a random deviate from a Gaussian distribution with variance, with mean zero and variance dt. But it's not written like this, never. So, so I don't think it's a proper way to say it. But basically, you're adding some Brownian noise here, and that makes you have some change over small time. You change by an expected amount. So every line will change by this quantity. And then each line will have its own, it will furthermore draw a random deviation up or down, which will be drawn randomly and which puts the noise in the demography. And this draw will be given by this. So it's really something that matches the mean and the variance of the change in population size over a small time. So that's why it's a diffusion approximation. From the whole demographic process, you summarize it by a mean change and a variance in the change, and no more. So there are several branching process that can be approximated by this. One of the classic one used in a stochastic rescue models is for discrete population. It's a branching process where you assume that the number of offspring from by one guy over one generation is drawn into some Poisson distribution again. I don't want to go in detail why it should. It's basically, I don't know. There are probably ways to derive it properly, that it's a Poisson from some assumptions, but usually it's really to have the simplest discrete time model of variable offspring number with just one parameter. So then the Poisson has a parameter Wi. I called it like this because it is the absolute fitness of the Darwinian fitness. So it's a W really, and this will be Malthusian fitness because it's the expected growth rate of the genotype. So it's the Malthusian fitness, and this is the Darwinian fitness. And then if you do that, basically sigma for every genotype I is 1 plus Ri. And if R is small compared to 1, which is required for the diffusion limit, so per unit time, then it's roughly 1, this. So you do have a constant sigma across types, roughly. And the mutation rate is U, common to everybody. And Ri is what we did. It's the log of the Poisson parameter. If you take a birth-death process, one way to have it is to have a birth-death process with just genotypic variation on the death rate. There are several ways to have it. So there are other ways to have it, but this one is simple. Then Ri is the birth minus the death rate. And the mutation rate is constant across type because you assume the birth rate is common to everybody. So you have a mutation rate per division times the birth rate. And sigma is B plus DI. And here you assume basically that the birth and the death rates, they vary between each other. They have a difference, but the sum of the two is large compared to the difference. So it means that basically your genotypic changes, they change the death rate, but not to a level that the birth rate originally is big anyway, and the death rate originally of your wild type are big too. Their differences are small, but they are both big. So that when you change them a little bit, both of them, you change a lot the growth rate from positive to negative, etc. But the sum of the two is not too much different from what it was in the wild type. So then in this case, sigma is also a constant. There are other ways to get the Feller diffusion as a limit. So I won't go into detail. That's what we assumed. In the simulations I will show, we use the discrete time model, so this thing. And for the moment, I don't think there are papers, at least our papers, we did not study the effect of variation in mutation rate and in sigma. Every time there is in gray down the thing here, there are references corresponding to where it's discussed. So this tool gives us other handy results that we can use. First, in the Feller model, we know the extinction probability at a given time. So at any time, we know the probability that a Feller process will be extinct. So in the notebook I will send, there is, for those who want to look into really the math, there is the derivation of this formula. But I clearly won't talk about it here. But we have a formula, an explicit formula, quite simple. So that means we have access to the probability of getting extinct. So we have access to the establishment probability over infinite time. And also for any line, because we start them in one copy, we know what the likelihood is that they get extinct. And we also have access to the extinction time distribution, because if we had, for example, a Feller process for the whole population, we know the probability of being extinct by time t. So it means we by deriving it, we have access to the distribution of the time to extinction in the population. There's another quantity that is a terminal variable that is also analytic. I won't talk about it too much. And another interesting property, because this we have it also for a birth-death process, for example. They are actually very close, the birth-death and the Feller diffusion results. But what we have in the Feller that we don't in the birth-death is we have access to the distribution of this quantity. So this is the cumulative size of a lineage from time 0 to its extinction. And that's very important when you deal with intermediate lineages, because when you do a springborn mutant, this mutant has a stochastic fate over time, and it's the cumulative distribution, it's the distribution of its cumulative size that tells you how many mutants this guy is producing. And this thing is very stochastic because you go from 1 copy to a 0 copy, and you go through a very noisy trajectory. So this, you cannot ignore the stochasticity in this. You cannot approximate that by an exponential decay clearly. Whereas we can do this for the exponential decay of the wild type. If N0 is large, you can actually prove it from this formula, which is the exact one. And you can see it makes almost no difference to consider exponential decay all the way to death versus the true stochastic cumulative number. But for the springborn mutant, for the 2-step mutant, it's very, very important to have this. And this is only available in continuous time approximation, continuous state approximations, I think. So not in the birth-death. Okay, so we have these different tools, so we can use them all that remains now is to plug the FGM results into those probabilities and to have access to the ER probe. So there are two ways to deal with the thing. The first way is in the SSWM, so strong selection, weak mutation. The genetics is simple because we assume that every rescue will be due to one mutant that will make it be established and make it to invade. It doesn't mean we don't consider all the mutants. It just means we are modeling mutation as the one mutant in every replicate. In every replicate, it will be a different mutant with different growth rates, but we don't need to deal with multiple mutations. And also in this regime, we can also consider that the standing variance contains at most one mutant that is in the standing variance. There's no contribution from double, triple mutants to rescue. So in this case, we have a simple genetic basis, but in exchange for that, we have to deal with a fully stochastic dynamics of this. In the other case, so this is then each lineage is modeled as a homogeneous failure process, so with a given Ri and sigma, and we can deal with single or double step ER. And the standing variance is like almost like the spike with some rare standing variance. So these are the two papers where we use that. And the other regime is when, on the contrary, we have a strong mutation and weak selection. So then the genetic basis is a mess very quickly. Well, first in the standing variance, there is a lot of polymorphism, and in the response, many mutants are produced all the time. So this then is a mess in terms of population genetics because there are many variants everywhere. But on the other hand, in exchange for that, this mess means that the trajectory of mean fitness of the population is more deterministic because there is a lot of input of mutants, which means that drift plays less of a role compared to mutation and selection. And so in this case, we will use this approximation, which is we consider evolution to be deterministic. So it's following the deterministic trajectory of mean fitness for that new environment. And we just will consider that demography is stochastic, so that it's just the demography, the noise around this expected mean fitness that creates the extinction or rescue. And then it is fundamentally an infinitely many step ER. And the standing variance in this case also is different from the other because it's the Gaussian case. So this is the other paper where we do that. Okay, so we don't have a treatment for the intermediate. I'm trying to do it right now, but it requires quite elaborate stochastic treatments. So first, the single step. So that's the strong selection, weak mutation regime. This is what looks the most like the population genetics we use here is the one we derived yesterday and the day before. Just we're plugging in the distribution of mutation effects and the cost distribution from the Fisher's model. And also we're plugging in the establishment probability from the failure diffusion in this case. Once we do that, there are still three things that can happen. So in early models of rescue, the ones with sexuals, what is considered typically is just a shift in the optimum, either continuous over time or abrupt. But in fact, even if you're considering that it's a simple quadratic fitness model, there are other things that can happen when you change the environment. First, you may also go down. The absolute maximum possible fitness in the new environment might be lower. And second, you may also change the variance that is available by mutations in the new environment. So you may have some mutants in the first environments that have a given variance, and mutation may produce a different variance in the other environment. In the isotropic model, it's completely equivalent to consider that you change the variance of the mutants and keep the quadratic fitness function the same, or that you keep the same variance and you change the quadratic fitness function. Basically, they all boil down to one parameter. So, and if the two happen, it boils down to one parameter still. So this covers the fact that maybe selection is stronger, for example, which is really the same as saying that lambda is smaller. Or that the growth rate, maximum possible growth rate goes down, so you can not do better than another R max, and that the optimum has shifted. So what is interesting is that under an approximate treatment that basically the mean fitness effect here is because that's connected to the average selection coefficient of random mutations. This thing is small compared to the maximum growth rate everywhere, even in the new environment. So this is really saying the growth rate itself is big compared to the mutation effects. If you are in there, then things simplify. All those scenarios, in fact, they boil down to a relationship between the probability of rescue and a single composite parameter. And this parameter is given here. I don't go into detail, but it's including both the decay rate imposed by your particular environmental shift, the change in Rmax, the Rmax you have in this new environment, and the change in lambda, the lambda you have in the new environment. So it's accumulating all them together. So PER, the probability of rescue, takes a form of 1 minus E minus something that depends on the conditions times N0. So that's expected all the time when you're considering density-independent models, because the fate of every single mutant is independent of the other. So extinction from N0 mutant is just extinction from one guy to the power N0. So it's always going to be like this. And then this rate of rescue per initial individual, if you want, is given by this formula. Oops, sorry. I don't know if you see my mouse when I'm showing things with the mouse. So basically WRDN, DN for de novo, that's the de novo part. It's given by this formula, linear with mutation rate and something that depends only on this composite parameter. The standing variance will not depend on RD, well, depends on RD, but not Rmax and lambda, because we're considering the past environment where the absolute fitness don't play a role. So it's a bit different. And here is an example with simulations versus the decay rate. So by decay rate here, we did it by shifting. We just shifted. We didn't make it go down or narrow it. And these are simulations, the dots and the analytic approximation. And if you add standing variance, you have another analytic approximation, which is using the costs that I'm not showing. And what happens is that when you allow for standing variance, basically what you do is you increase the range of decay rates where you can rescue. So you see here, beyond 0.3, there was no rescue, or beyond 0.2, there was almost no rescue. With standing variance added and the same mutation rate before and after, you have shifted your range of environments to which you have a substantial probability to resist. But otherwise, the shape is the same. Okay, so we have analytic explicit expressions for that. Now, in the same model, we can also derive the two-step rescue. So it's a bit more tricky because, as we said, the two-step depends on having this intermediate springboard that is going up and down very quickly. But these are fundamental because they are the guys who produce the second resistance. So you need to model them. And these guys, these intermediate guys, may be at different places in the landscape. You start from someone out of the, so I'm going to show it, but they may be different phenotypes, those guys. But first, the pattern. So here is, so again, here you have an analytic expression for the probability of rescue in 1, actually 2, 3, 4 steps, but they are ugly numerical integrals. Huh? The two-step, I think Matthew had, so that's paper by Matthew Osmond, had some approximate expression. So anyway, in black, this is the full rescue probability versus the wild type growth rate, so or decay rate, but with a minus. So of course, as the decay rate increases, you have less and less rescue. And these, so again, dots are simulations and lines are analytic approximation. What is important here to see is that the blue is the part of the rescues that are done by one-step rescue. And the red is the ones by two-step. And then the brown is 3-step, 4-step. This is log scale probability of rescue. So what you can see here is that as long as your decay rate, your stress level is not too large, most rescue come from one step. All the time at all growth rates, rescues are mainly done by 1 or 2 steps, meaning that those lines, those 2, 4 steps, they're always some log below the 1 or 2 step, at least the 2 step. So the 3 and 4 don't need to be followed, at least in this parameter range that we use. What is important also is that the 2 step, they take over for very large decay. So when you have a strong stress, almost all the rescue comes from 2 steps now, because it's too hard basically to produce a resistance to by 1 step. So that's a bit like when you have very high drug doses. Sometimes you obtain it empirically by selecting your bacteria on an intermediate dose and then putting them in the other dose, and they accumulate two mutations. Here is the phenotypic place where those steps, those 1 and 2 steps are. So here we're seeing the landscape from above. We're looking at phenotype space with 2 traits in this case. The optimum is at the center of the target. And the blue is the wild type. It's standing at some distance from the optimum because it's in the stress. And the gray circle is the area where you have a positive growth rate. Whenever you're outside of the gray circle, of the black circle, you're subcritical, meaning that you're decaying. Your growth rate is negative. When you're inside the white area here, you are supercritical, you're growing. And when you're in the gray area, it's the critical zone. Basically, you're neither growing nor decaying. You're in between the 2 situations. So the 1-step rescue is starting from the wild type, boom, you jump inside the growing, the supercritical area. The 2-step rescue, there are 3 ways to have it. The first one is to mutate to something that is a bit less critical, subcritical. So basically, this intermediate mutant is still not a resistant, but it's a bit better than the wild type. And first, because it's better, it's decaying less fast. And second, and that's really due to the landscape geometry imposing this, because it's closer to the optimum, it also has much more mutants that can make it to the white zone. So this is why it's a springboard. The other type of springboard that has a different behavior is guys that are critical. So they are jumping to the intermediate line. And it's different for a reason, is because those guys, they can maintain themselves as a flat line for a long time. So the production of random mutants is different from guys that decay in a little time. And the last case is supercritical mutants that are springboard. So they are like this because they could have been first step rescue because they are resistant, but we condition them on being those first step resistant that do not establish. So they will be lost anyway, so they don't make a rescue. But before they get lost, they may produce a second step that will be a rescue. So those are the last type of rescue. And the proportion of each type depends on the stress level. So as you increase the stress level this way, here is the percentage of types of 2-step rescues. And you can see that the subcritical 2-step becomes dominant for strong stress because basically you're so far away here that you cannot make the subcritical or the supercritical case. You always end up, most of your mutations end up being not resistant in 1 step. So that's why your subcritical springboard are dominant. Then the critical is roughly, well, the critical and supercritical are roughly doing a bit the same, and they are less and less contributing. If you look at that same genetic basis of the 2-step rescue, but depending now on mutation rates, then you have a different trend, which is that as you increase mutation rate to high values, these are, again, the dots are simulations. The lines are analytic approx. Then for low mutation rates, most of your rescues, they are either subcritical guys or supercritical guys. But for an increase in mutation rate, you start to have more contribution from critical guys. The reason is that, well, what interpretation we're doing with hands of this is that the critical intermediates, they stay for a very long time. So if you have a lot of mutations, those guys have an advantage compared to the others because as they stay for a long time, they produce mutants proportionally to the mutation rate and in a large amount. So their advantage of staying a long time becomes more and more strong when there are many mutations because they produce many of those during this long time that they spend wandering around. Okay, well, this is really with hands interpretation. The result is just what you see. Sorry, there's a question.
Yeah, I was wondering why this benefit of the higher mutation rate is not seen for the supercritical ones.
So I suspect, but again, it's always a tricky exercise. I tend to not like it when you have a result that is just a mathematical result. Then you check it with the simulation. Okay, it seems to work. And then you try to interpret what happens with the hands, which is important, of course. But that's where you're not rigorous because you don't know if your interpretation is correct. So one interpretation I would have is that the supercritical guys, their contribution to two-step rescue becomes less as the mutation rate increases, just because most of them become single-step rescues.
Oh, I see. But in the
simulation, you produce a lot of those, and they just are single steps.
Okay, so are you not able to tease that apart in the simulations results?
Yeah, we probably are. We just didn't do it, I guess. I also have to reread the discussion of our paper because maybe we discussed that in the paper and I don't remember it.
I mean, if they were single step, then they shouldn't get counted in your.
The single steps here are not counted because we're just dealing with the sum of the percentages here must sum up to 1. So it's all the single step, the two-step rescues.
Right. So then what you just said would not work, right? For explaining what this trend is, because you're not even considering the single steps because they would already be outside of.
Yeah, but I'm thinking maybe it's wrong that because they're contributing more to first step, I need something different. I need that the supercritical contribute more relatively to the other guys, to the other two-step intermediates to rescue, which I don't know. So yeah, I can't answer. Maybe it's in the discussion of the paper.
Okay.
Like that, I don't have a this pattern. I don't remember how we discussed it. I don't know how we discussed it. So it's
okay, I can look at the
far
paper.
away. Sorry, but it's I hope it's going to be discussed in the paper. Yeah. This pattern is more easy to understand because you can see that when you're so far from the optimum, most of what you manage to do is to jump to some still subcritical intermediate. But this pattern is less obvious to me. But yeah. I'm gonna go to the last case because now the last modeling is when we change the parameter range and we go to even higher mutation rates. So U over lambda becoming more and more bigger and bigger. And in that case, the way we model it is very different because what we do, so maybe I should skip back to the slides for the methods we use. So here, what we do is really different. We use a deterministic evolution, but we consider time inhomogeneous demography. So if you get back to the equation for Feller, basically what we do to derive the extinction probability then is instead of considering each RI that is fixed over time, but there are many RIs and we deal with each individually. Basically, we are dealing with U of R and RD, but we're just integrating over the distribution produced by the Fisher model. This time it's different. We consider DN, the whole population, and it has an SDE. And this SDE now, instead of being given by, it's given by R bar of T times N, which is really what the expected, this is really the exponential growth rate of the population at any time, and plus sigma, which is constant, times N of T. So by doing that, what we do is we say that the R bar here, it must be a deterministic function of time for the treatment. And so by saying it's a deterministic function of time, we're saying that the population evolves with the mean fitness that is given by the infinite population theory, and that the demography is still stochastic, but with a mean fitness that is fixed. So there's no, in our paper, there's no explicit justification of that. It's just we're going to check this option. The kind of intuitive justification is to say two things. First, n times u is large. So the population size, initially at least, has a lot of mutants flowing in. So it has a deterministic trajectory, at least initially. And as you get closer and closer to the extinction, this is not true anymore, of course. But the thing is that a lot of the fate of the population is played when you go fast enough to growth rate 0, which is where your population size will rise up again. And the error you make later on is less important, if you want. But there's no rigorous justification of that. We're just taking this option to see what it does. There is, the only kind of intuitive is to say, what is critical is this initial adaptation, which makes you avoid, get extinct at a slower pace. And then when you're close to extinction, it's the noise of your demography alone that makes you hit the boundary of 0 or not. That's the origin of the approximation, if you want. There's no rigorous demonstration of it at all. But yet, in spite of that, it does a good job in the parameter range where the other does not. Because, of course, you can see how we cannot go on and on adding multiple steps, because every step implies an integral on an integral on an integral. So analytically, we have nothing that we can do with too many steps. And when the mutation rate becomes large, these multiple steps become substantial. And so that's what we get then. So here, for example, we took two extremes to illustrate it. You have a mutation rate very low, way beyond lambda, and a mutation rate very high, way above lambda. And you can see how in this regime, you have the theory for the weak mutation, the WSSM is correct. So the dashed line is the SSWM, so the strong selection theory, and it's correct with the strong selection simulations. And the weak selection is wrong. In the other case, it's the opposite. When you increase U to be large, of course your single step rescue is wrong, and your WSSM is correct. I did not plot the two-step case because the paper is actually anterior to the two-step paper with Matthew Osmond, so we didn't have the theory for that. But the basic idea is to say that those two things give the same robust pattern, that there is always a very sharp decay with RD, and that this decay is not due to just what we saw in the population genetics of it, which is that the rate of rescue is in 1/RD, just due to the fact that you have less mutants being produced when RD is bigger. This drops much faster than that. And the reason, in both cases, intuitively at least, we believe the reason is that RD is not just the number of guys you manage to do as a wild type before you die out. RD is also telling you how far the optimum is. And that does a huge change. You can change a little bit the distance and have log scale decays in probability of producing a rescue mutant. And this is because when you're dealing with rescue, you're not dealing with classic mild changes in the environment. You're dealing with cases where your shift is so large that there are only a few mutants that make it. Otherwise, rescue is probability 1 and you don't study it. So in this area, what counts is really the tail of the Gaussian distribution of mutants. And this tail drops off at some pace where a small change, it's really due to the tail of the distribution. A small change means a strong change in the probability of having a resistant mutant, and therefore an established mutant too. Also, there is something else arising when you start to deal with these large mutation rates. You can show that if you plot stress level in log scale versus the decay in PR, you can see that this thing is not a straight line. There's a sharp change like that. If it was just U over RD, proportional to U over RD, we would have a straight line with slope -1 here in log scale. So something looking more like this. So it's really different from just the RD effect from the demographic, purely demographic effect. And also putting standing variance, which you can do also analytically, just shifts the probability of rescue. Again, same as the previous model. Standing variance just increases the range of environments to which you may adapt, to which you may survive. But the shape is the same in log scale. Another important thing is that if you plot rescue versus mutation rate now, you have a different outcome, because as you increase mutation rate, of course, you increase rescue probability. There's more rescue available. It does not increase proportionally to mutation rate as it did for the single step, of course, but it increases and then it plateaus. But when you reach a higher and higher mutation rates, all of this drops back down to 0. And that you can only see using a weak selection, strong mutation model, because this is just the mutation load now that is killing the population, because it's so high that in spite of being able to withstand the shift in the environment, at the same time, you're loading yourself so much with mutations that you're killing yourself anyway. So you're going from really rescue here to what is sometimes called lethal mutagenesis here, so which is basically getting killed by your own mutation rate. And so this appears only in this kind of model because it has the effect of buildup of the genetic load in the model. Also, because here we have just a single Feller diffusion, inhomogeneous, we can work out the distribution of the times to extinction, which we couldn't. So because we can work out the probability of being extinct by some time t. So here, this is the distribution in the simulations, and the red line is the explicit formulas. And so here is for various mutation rates. So here, the darker means higher mutation rates. Umax is the threshold where you have lethal mutagenesis. And you can see that as you increase the mutation rate, your time to extinction gets more and more spread. So you have some lines getting extinct after some time and some others after another. The threshold here is just the minimum time it takes to get extinct, even for the wild type without mutation. Now, if you change from Umax to infinity, so that means now you're in a regime where extinction is certain and it will happen due to lethal mutagenesis. Here too, you can work out the distribution of time it takes you to kill yourself by lethal mutagenesis in the presence of the stress. And this time it goes the other way around. So the distribution is spread when you're next to Umax, but as you increases further away, you get a narrower and faster extinction, because of course lethal mutagenesis is faster as you go to higher mutation rates. And the last, of course, is depending on the decay rate, so the strength of the stress. So here, as you increase the decay rate, extinction happens faster, and it's more narrowly distributed. So this lethal mutagenesis effect has a last implication, is that the threshold at which you kill yourself depends, well, let's say, no, it's not this. The stress level you can resist, if you increase the stress level, you require higher and higher mutation rate to withstand it from a purely rescue theory point of view. But as you do that, at some point, you need a high mutation rate to manage the environmental change. But this mutation rate is so high that you kill yourself. And so in some parameter range, there is some level of stress that you cannot overcome anyway, whatever your mutation rate. So to illustrate this is a plot of stress level versus mutation rate. And it's a density plot. So the more white, the more probability of rescue. So the blue area means there's no rescue possible, and the white means rescue is always happening. And so you can see here that this is for two different parameters. You can see that there is, so this is a sharp analytic, this is like an analytic threshold we derived for where this area is. These are the conditions where you can do rescue, the conditions of mutation rate and stress levels. And there are some areas of parameters that you cannot do rescue whatsoever, because basically to be able to overcome the environmental shift, you need mutation rates that are imposing you to be in the lethal mutagenesis, so you don't do it anyway. So that's interesting, more or less, in the idea of having, of course, treatments that you cannot overcome. The limit of it is that I think that they require mutation rates that are very, very high. So basically, the idea is you treat both with a mutagen and with a treatment. The problem is that in between, the mutagen is helping your guys. So if you don't dose your mutagen right or your treatment right, you're actually killing your patient rather than helping. So it's not maybe the best way to do. So I'm almost finished. Some robust results on all these rescue probabilities in Fisher's model. First of them, which would happen in many other models, is that the ER probability takes this form. So 1 minus exponential minus n0 times something that depends on the parameters and the environment. But that's just saying that any model with density independent processes and mutation and no interaction between lineages, so no sex and no density dependence, will generate that just because the fate of each individual lineage is independent. The rate of rescue is going to be proportional to the genomic mutation rate, only for strong selection weak mutation, one step, not otherwise. It's going to be more responding less than linearly with mutation rate otherwise. What is happening all the time, and that I think is important, is that there is a very sharp decay in the rescue rate versus RD. So a small increase in stress in terms of decay rate means a strong decrease in rescue probability. And that's important for treatment views. It means that it pays to increase the stress level by a little bit. It pays a lot. Well, the genetic basis, it ranges, well, from single to 2 to many steps, depending on those ratios. And basically, in both very polymorphic or low polymorphism models, what happens by authorizing standing variants to be there, at least at mutation selection balance, that's the one we studied, is just, you don't change the shape of all that, you just shift, you just allow the population to survive more higher stresses, or to survive the same stress at the lower mutation rate, put differently. Now, just a quick illustration on data. This is experiments that are done quite often in microbiology when dealing with antibiotics, drugs in general. What they do is they look at different drug doses that they vary by log scales in concentration, and they take a population of bacteria, white type bacteria, which is actually polymorphic, and they plate this population on plates with different log concentrations, and they look at how many colonies they retrieve from that. So in principle, although it's not a proper fluctuation test because you have covariance between your different platings because you grow a single culture, whereas a fluctuation test you need to grow individual cultures for every replicate so that they don't share common mutations that arose in the beginning. But it still gives you a good order of magnitude of how likely it is that you produce a resistance mutation, actually a rescue mutation, because as we saw, producing a colony means avoiding stochastic loss when you're in a few cells. And you can see here that the rescue probability, so this is really log PFU per plate, so it's a log scale, it's a proportional, it's a constant plus log rescue probability. It drops very sharply for different lines here, and for different species, in this case, with the drug dose. And the same happens with different, I think this one is with different fluoroquinolones, different types of the same class of antibiotics with the strain. And these really look like they are log-log. So this aspect that decay rate, well, let's say more precisely, drug dose is not decay rate. Our model says that probability of rescue decays log-log linearly with decay rate. These are probability of rescue with drug dose. And at some point, indeed, drug dose and decay rate, there is a sharp change in drug dose versus decay rate, roughly proportional, but well, yeah, but not everywhere. So it's basically in typical kill curve experiments, there is an area where it's linear and then it flattens. But it does go in the idea that indeed there is more loss in rescue priority with stress than just a mere demographic effect of 1 over the decay rate, just a mere demographic effect. Plus, if you consider that standing variance is an important component of the rescue, which is in several models is the case, we do not expect any effect of RD on the pure population genetics model, because the model depends not on RD, it depends on what happens in the past. So it depends on the mutation rate to resistance. That's all. So this suggests that indeed there is an effect to take into account, that the probability of mutating to resistance drops sharply as you increase the stress level. And that's an important thing because to me, at least, because there is a discussion, well, first for handling treatment, but there is currently a discussion of what is called the competitive release effect, where it's said that if you put intermediate drug dose, you will help the treatment, because intermediate drug dose, they kill the wild type, but not too much. And so the wild type continues to hinder the growth of the mutants that are there and that will invade. And those theories, in fact, they don't model the mutation rate of the standing variants. They say that it's the same whether the drug dose is high or low. And so, of course, everything that happens is due to the density dependence release. By killing less the wild types, you let those wild types hinder the growth of the mutants. But if you start to put in a very important ingredient, which is that drug dose means that there are much less resistant mutants in the beginning with a higher drug dose, then it changes a lot. And also in the experiments that are done to test it, what they do, in fact, at least the ones I've read, is that they put half of the population with the wild type and half with the guy who resists all drug doses. So you're creating an initial state that never exists. In fact, for free, you're putting half the population resistant to a huge drug dose. Actually, when we want to produce resistant mutants to a high drug dose, we do put them in an intermediate dose to produce them. We screen on first an intermediate dose, and then we screen to a higher dose. So the intermediate dose regime is exactly the one to produce super mutants that are resisting any dose that you cannot kill anymore at all. So I think it's an important thing to say. That's and last, here is experiments that I'm doing in the lab with bacteria. So it's E. coli, and they are transformed genetically to express luminescence. They also express fluorescence, but here it's luminescence that I'm showing. So what is luminescence? It's just what allows bacteria or medusa, jellyfish, to produce light, or firefly. This makes them use energy and produce light. So bacteria can be transformed with the called the Lux operon, which is a bunch of genes that if you insert them in the chromosome or the plasmid of your bacteria, they constitutively produce light. So compared to the eukaryotic Lux gene, the advantage of this one is that it's autonomous. You don't need to add the substrate of the reaction. It's producing the substrate for the reaction and producing the light. So basically, the bottom line is that a bacteria that is currently metabolically active and not lysed and has a not compromised membrane produces light at a quite stationary level when you're in the stationary physiological state, which is basically the stable edge distribution regime, which is the exponential phase. So that allows to follow. It has high signal over noise ratio because it's light produced. It's much more sensitive than optical density. And also it detects some death that you don't detect with OD. OD, you detect death when the death is imposed by a lysis of the cell. exploding. So with phages, it works fine. With ampicillin, actually, which is the antibiotic I have, which are penicillins, it works also quite nice because it explodes the cell usually, but it's much less sensitive. So that's the reason we're using this Lux transformed guys. And so what we do is at some time 0, so this is Lux signal, so proportional to the number of live cells. At time 0, we put an antibiotic at a given dose. These are 3 doses, so the control in green. All this here, the top dynamics is just wiggly things happening when you reach stationary phase as a bacterial cell. The luminescence per cell changes because the metabolic activity shifts actually in a very repeatable manner. But we're not interested in this area because we're dealing really with below stationary phase dynamics. And also we know it is because the fluorescence does show a classic logistic curve, theta logistic curve. Anyway, more important is what happens to those 2 treatments. So this is with a high dose of 4.6, and this is it's a lower dose by just factor 3. And so you can see what happens is that first, there is some time, which is really a long time. You have to realize the time it takes before it starts to drop. Then the drop occurs, and then rescue occurs or not. So rescue, in the case of the high dose, there's 1 rescue here among those replicates. And in the case of the lower dose, we have quite many rescues. And you can see several things. So first, of course, the probability of rescue drops with the dose, and which corresponds to faster decay rate. So that's what we have in mind. Second, there is a distribution of rescue mutations. So you can't see it with this one because there's just one, but with these, you can see different growth rates here. But another thing that is not in the rescue models most of the time is this, that in fact, even when you impose in a test tube, in a microplate here, fixed environment, not a huge population, so and you impose a given concentration of your drug, the environment is not a fixed environmental shift. There's a time delay between the time it starts to act and kill and the time you've put it in. So in fact, what these cells are undergoing is a changing environment, continuously changing environment, more or less, or maybe discretely in 2 steps. So there are models that we're doing with this for this phase, just a demographic mechanistic models. But it's important to see that even in simple lab conditions, it's actually difficult to impose an abrupt shift. So it means it's an advocacy for continuous time changes in stress levels. And the other thing you can see here, well, I said it, it's that there is variation in ER mutants. So that it makes sense to try to consider distributions of resistant mutations. And also that there is not one, when we say this bacteria is resistant to this drug, it's of course meaningless. It's resistant to this drug dose. And there are others that's obvious. But, and it's just to show that you can also have access to those dynamics and try to test models with it. And I'm finished in advance for once. So.
Yeah, thanks, Guillaume. I just have a general question here. I mean, not really, I mean, so on one hand, we have this mutational meltdown models, right? In which, because
it's not mutation meltdown. It's
I understand.
lethal mutagenesis. It's very different.
No, I'm not talking about lethal mutation. I mean, I'm just saying that demography, how it changes. It could be just loss of the population because of mutational meltdown. On one hand, we have that limit.
So the Müller's clique, Müller's ratchet applied. Müller's ratchet with demography.
Müller's ratchet, correct. For asexual populations, right? That's right. Yeah. On the other hand, you are considering asexual evolutionary rescue. So I was wondering. I mean, shouldn't the more general model have both deleterious and beneficial mutations? I mean, I believe you have only beneficial mutations, right? Because you're interested in rescuing them.
They're everywhere. No, they're. No, it's important. I need to get back to the Fisher's model. Well, you can see it on the clouds here. This is the wild type.
Mm-hmm.
every mutant, and this is the random mutations produced. Every mutant that is outside of this latitudinal level is a deleterious mutation. And each of those are beneficial mutations. And among those, some are resistant mutations.
Okay.
And among those, the blue ones are resistant mutations who established, whereas the red ones are resistance mutations who did not establish. That's why they are red.
Okay, so there are.
But everything below that is a deleterious mutation. Okay. That's the point of the Fisher's model. It has all deleterious and beneficial mutations all together in one cloud, and it has also epistasis. That's very important. Talking about mutational meltdown. Mutational meltdown is Muller's redshift, and so it makes me crazy how it's been studied. Whereas biologically, to me, I don't see any situation where it has a meaning, because to have mutational meltdown, you need to have non-compensatory evolution. There must be no compensation by mutation, so that every time you click, you did click really. So that's the first reason. We know compensation is everywhere. So okay, but then the other thing is that the time scale of the meltdown is the time scale of the click. And the time scale of the click is the waiting time to lose a given fitness class to go to the next fitness class in the deleterious ranges, right? And this next fitness class with a distribution of mutation effects, it's just an infinitesimal point away from the first one. You see what I mean?
Yeah.
And it takes you log n time to fix it. And by log n time, you've clicked by, say, 0.001%, and you need to click again. So when you look at papers with mutational meltdown, you can look at the burn-in times that they use, the time scale that they use. These are eons of generations. So it means that the mutational meltdown is a way to get a population extinct. To me, maybe I'm too extreme on it. I've looked a bit on the theory that goes with it. It takes very long. First, it disappears whenever you allow for any type of compensation. So of course, in Fisher's model, you have compensation, because if you're deleterious here, you click, so your population fixes for this. You have all the mutations above you here that can make you go back up, so you'd never click, of course. That's a paper by Otto and Poon, who showed that compensation just kills out the meltdown. But the other thing that is often not realized, I think the papers I've read have that, is that the time frame of mutational meltdown is by factors of log n, because you need a log n to click to the next less fit class. And the next less fit class, when you have a distribution of mutation effects, is a very small one. Rigorously, it's an infinitesimal class. So it's random. So you will click sometimes to a less fit class 1, sometimes to another less fit class 2. But these are like within percents. And to click to them, you need to lose a whole population, because you need to lose the whole. Actually, you can compute the size of the population itself. It's in a non-epistatic model. It's going to be the same roughly as this. It's E minus U over some harmonic mean of the selection coefficients. And so it's in E minus U. That's the frequency of your big perfect class, and you have to multiply this by the size of the population. So this thing is very, very large. The number of individuals at the fit class is very large, and so you need to lose completely this very large class, and so it takes very long time to lose them. The order of magnitude of neutral fixation, but it's not even this. It's I think it's bigger. And then once you've done that, you've clicked by 0.1% of your previous fitness. So basically, you've clicked, there's nothing. And you need to wait for another eons of time to click to the next one. And that's why in the papers doing Muller's redshift, you can look at the burn-in times they use. They use like 10 to the 6th generation burn-in time before they check their theory. So basically, they're telling you something about what happens in 10 million generations away from now, which is, I mean, never useful. So to me, it's a very fun, nice probability exercise. It's pretty useful in the tools it's been developed due to this. But my impression, at least, is that it does not describe anything that happens in real populations, just because there is compensation, and that means there's no click, and because the time frame of those decays of this meltdown is very, very long. So that's an impression. Again, I'm not expert on this, so just check what I'm saying is wrong or not. It's just
my question was just that, whether there are both beneficial and deleterious mutations.
Yeah, there are both. Yeah,
both
and
are,
they are
right?
epistatic. They are epistatic.
Yeah,
so
right.
There is a pervasive epistasis in the model.
Yeah. There's some interesting stuff here, but I mean, I think I'll have to have a look. I got lost, so I think
if you got lost, I can't imagine the students.
Okay, I think many of them have left. no, I just said I think I just need to have a look. Yeah. Any questions? Anything? Okay. So I don't see any questions. So anyway, Guillaume, it was nice having you at least online here. So, and you told us about interesting new things. So it was fun. And let's hope to have you sometime here in person. Okay? Yeah. Have a nice
Thanks. I hope I didn't disgust you to invite me in real.
No, we were not
sure.
Pushy. Don't worry. We were not pushy.
I can change the contents of
it's
the
just I think we'll have to sort of
so
think.
please feedback also the students on what was bad on it, because it's useful for me.
Yeah, I think it was all good.
It's anonymous, so they can say whatever they want. I don't know them, and I'm 1,000 kilometers away, so I can't harm them anyway.
So I think it's second week, so people are generally tired, so I don't think it's a reflection on you or anything, so don't worry about it. Yeah. Okay.
Okay.
Take care.
Thank you.
Thank you. Goodbye.
Bye.